All right, good morning to all of you. So I had started with all of you before the midterm exam, and let's continue on it further from now onwards. So the topic for today is protein degradation, meaning protein breakdown, and intracellular protein degradation. Intra means inside and cellular means cell. So inside the cell, how proteins are degraded. Now by now you have a fair idea that most of the structural component as well as functional component of a cell is proteins, correct? You have seen molecular mechanisms where transcriptional factors are involved and enzymes are involved, and you have seen the cellular structure also, the membrane and all, where the protein is involved. So largely it is protein. Now most of those proteins, they are made, they do their function and they need to be broken down. Or there might be some proteins whose life cycle or the time span is complete and they need to be replaced by a new protein. So the old one needs to be degraded so that it can be replaced by a new one, and that is why we have protein degradation mechanism. So if I give you an overview of why you want to or why you should know it, is with the help of protein degradation, the cell is able to maintain a constant supply of amino acids. Again, you have gone through translation, you know that proteins are made from amino acid. So in order to make new proteins, you need to have a source of amino acid. So that is one of the functions of protein degradation. Another is that it prevents accumulation of abnormal proteins in the cell. So any protein that is old, any protein that is damaged, any protein that has completed its work, it should not stay back in the cell, because if it stays back in the cell, it becomes more and more. As a result, the environment of the cell becomes toxic, and when the cell becomes toxic, it spreads that toxicity to other cells, and that can give origin to a disease. And the cell does not want that. So that is also one of the reasons why protein degradation happens. Now, how do we define protein degradation? Let me read this sentence and then I'll explain it to you. So turnover of a protein is not constant or same for all proteins, meaning there is a huge variation in the half-lives of individual proteins, which is the direct reflection of their role within the cell. So turnover of a protein or half-life of a protein simply means the whole lifespan of one protein. So this sentence is trying to help you understand that there are so many different proteins, their life or turnover is not same. Protein 1, protein 2, they have different lifespans, and that is a direct reflection of what kind of a role they are playing inside the cell. If it is an enzyme, for example, it will do its work and it will get degraded and it will get made again whenever needed. However, on the other hand, if you have a protein that is part of the structure, for example, peripheral proteins on your membrane, they are part of the structure. So they stay with the cell as long as the lifespan of that cell. Clear? Now normally or generally speaking, most of the proteins, their lifespan is 100 to 200 hours. That's the general time period for most of the proteins. However, There are some proteins which are short-lived proteins, and then there are some proteins which are long-lived proteins, which means they have lifespan more than this, and short-lived, they have a lifespan less than this. So under short-lived proteins, I have stated some examples here. For example, regulatory proteins. I give you an example for regulation of gene expression, regulation of transcription. In all those processes, certain proteins are involved. They work for that process and then they get degraded. Similarly with enzymes, enzymes that are playing part in a pathway, they will do that reaction and they get degraded, and so on. On the other hand, long-lived proteins are largely those proteins which are part of the structure of the cell. For example, crystallin. Crystallin is a type of protein that is present in our eyes, and I most probably now or in later years you will be told that eye is the only organ that does not grow after birth. So whatever is our eye size, largely it stays the same for life. So whatever protein, like crystalline is a part of our eyes, eye cells, retinocytes, it stays with the eyes until we die. So yeah, I am giving you two different examples, two extremes, okay. Also protein degradation, it depends on different factors. Now this slide is just an example to further help you understand that how protein degradation, the rule is not same for every protein. So for example, protein degradation may depend on the tissue distribution. I have taken an example of an enzyme, lactic acid dehydrogenase enzyme. Again, you will come across this enzyme in later semesters. So if we look at this enzyme, it is present in all the major tissues, whether you talk of heart, you talk of muscle, or you talk of liver. But if you see the half-life is different. In the heart, it is only 1.6 days, whereas in the muscle, it is the longest, 31 days, and in liver, 16 days. And that depends on how much that enzyme is getting used. So lactic acid dehydrogenase is used to its maximum in the muscle. That is why once it is made, it stays for 31 days and then get replaced by another lactic acid dehydrogenase. So you understand, depending on how long the function is for that protein, the lifespan is determined. Also, protein degradation is a regulated process. It's highly controlled and it can be controlled by our physiological condition. An example is acetyl-CoA carboxylase. Again, you will come across this enzyme in the next semester. It results in the formation of fats in our cells. Okay. Now, you know there are two physiological states. Actually, there are more, but the common ones on daily basis. We have a fasting state where we have not eaten anything for long hours, and we have a well-fed state that comes after we have had a good meal, correct? Now, fats are made when we have had a good meal, we are in a well-fed state. So this enzyme has its half-life of 48 hours when we are in a well-fed state. However, because in fasting there are not enough molecules to make fats, so the half-life of this enzyme reduces when we are in a fasting state. It is only 18 hours. Okay? So depending on whether we are fed or we have not eaten, that determines the lifespan of the enzyme. Remember it as a concept that depending on how much the function is there for that protein, it determines for how long that protein will stay in the cell. Okay? Now let's move on to the degradation mechanisms or the process or the machinery, whatever you want to call it. Now the proteins are degraded in our cells by two mechanisms. One mechanism you already know, it is lysosomes. It is a membrane covered organelle filled with enzymes having an acidic environment, and it breaks down proteins and other biomolecules also, correct? However, lysosomes can only break down 10 to 20% of extracellular proteins or intracellular proteins. So the amount of degradation that is carried out by lysosomes, it only covers 10 to 20%. So largely the protein degradation inside the cell is not carried out by lysosomes. It is carried by a different mechanism which is called as ubiquitin dependent proteasome system. And here I have mentioned it is in the cytosol. So this is like a machine which is present in our cytosol that breaks down almost 80 to 90% of our intracellular proteins. So most of the proteins that are present in our cells are not broken down by lysosomes, rather they are broken down by ubiquitin dependent proteasome system. Okay? Any questions until here? Since we are talking about protein degradation and lysosome is one of the component, let's quickly recap on it. So as you all know, lysosomes are organelle which are also like breaking down machines, correct? They are filled with digestive enzymes. As a class, those digestive enzymes are known as acid hydrolases. But under acid hydrolases, there will be lipases acting on lipids. There will be nucleases acting on DNA and RNA. And then there are proteases that act on proteins. And all of you know that the pH of lysosome is acidic. And how does it work? If you look at the illustration, for example, they have shown here that there is an mitochondria and it has become old. Now this mitochondria has to be replaced by a new mitochondria. And for a new mitochondria to come into picture, this old one needs to be degraded. So for its degradation, the membrane of the ER, it forms a vesicle around this old mitochondria, and when this vesicle is formed around the mitochondria, it is called as autophagosome. So remember, the vesicle that is formed around anything that needs to be degraded by lysosome, it is called as autophagosome. And from where that membrane is coming, it is coming from ER. This is the ER. Okay. Now when autophagosome has been formed, at the same time, lysosomes carrying proteins, it comes in contact with the autophagosome, they merge together, and the enzymes of the lysosome, they break the mitochondria and release the components into the cell. This I already have explained in detail in the very first resource that I took about lysosomes. So lysosome, it comes with the enzyme, it fuses with this autophagosome. The enzymes break the mitochondria, for example, in this case, and protein and other biomolecules, they are released in the cell. So this is how lysosome works. However, this is not the main agenda of this resource. The main is to know how proteasome works, how the main machinery works. So what is proteasome? Proteasome is an ATP dependent pathway, so this requires energy. Remember, lysosomes do not require energy. But if you are using something that is much more efficient and advanced, it requires ATP, and it is the proteasome system that requires ATP, and it is responsible for the breakdown of most of the short-lived, please remember this is the key word, most of the short-lived proteins in human cells. So most of the short-lived proteins are broken down by proteasome. And an example of short-lived protein is you have taken microfilaments, especially actin and myosin in the muscle cell, right? They are short-lived proteins. They will come, they will cause the contraction, and when the function is done, they get disintegrated. So that disintegration or that breakdown is brought not by lysosome but by the proteasome. Okay? Also, because proteasome can break down proteins, it can control many major biological events that are happening in the cell. So proteasome can control how the cell cycle will happen. It can control the transcription. It can control cell development and so on, signal transduction. And how does it control? It can control by breakdown of specific proteins that are involved in the process. Because out of all these processes, you know transcription, correct? And transcription happens only when transcription factors are there, right? So proteasome, for example, transcription happens. Once it is done, there is no need for those transcriptional factors. So what happens? Proteasome break those transcriptional factors down, transcriptional stop. If those transcriptional factors are not broken down at the right time, it will keep on happening and happening and happening, resulting in an abnormality. So you understand, in order to control transcription to be done for a certain extent, proteasome needs to break down those transcriptional factors. And similarly, there are so many, you might not understand these terms right now, but you will understand in the coming resources. Like, the signal transmission also involves proteins. So once the signal has been received, those proteins need to be broken down. If they are not broken down, unnecessary signal transmission is happening. It's not needed, but it is happening. Okay? Now, how do the knows that I'm going to degrade this protein, I'm going to degrade this protein, but I will not degrade this protein? How does it differentiate? There has to be something on top of it, something to show that okay, you can break down this protein, but you cannot break down this protein. So how are proteins selected for degradation? There is another protein which is called as ubiquitin. This ubiquitin acts like a tag, very good. It acts like a tag on the protein which needs to undergo degradation. So protein that are destined for degradation, we call them as substrate. So any protein that is destined for degradation needs to be tagged by covalent attachment of ubiquitin. Only then it will be degraded by a proteasome. So if it does not have a ubiquitin, the protein will not degrade it. This has ubiquitin, okay. So proteasome says, okay, I recognize you, I will degrade you. Okay. So since ubiquitin is so important, we need to know what it is. So it is a highly conserved protein. When I say conserved, it means that during the course of evolution, bacterias and then eukaryotes, and in eukaryotes there are so many different species until Homo sapiens came in. This protein has not changed much. If you pick up a bacteria and you take out ubiquitin, and you take one of your cell and you take out ubiquitin and you see them, you will find their structure more or less same. So it is a highly conserved protein. Has 76 amino acid, number is not important. This special characteristic is important, that on the C terminal of ubiquitin, there is a glycine amino acid. So on ubiquitin, all proteins have an N terminal and they have a C terminal, correct? So on ubiquitin, on the C terminal, it has glycine residue which is able to bond with a lysine residue on the substrate. Substrate meaning the protein that needs to be degraded. So glycine on ubiquitin, lysine on the protein that needs to be degraded, and they get bonded. And ubiquitin can be attached in mono or poly. Mono means one. So ubiquitin can be attached to the substrate, meaning the protein that needs to be degraded, as one, so monoubiquitinated, or as many, poly. And the process involves action of three enzymes, E1, E2 and E3. So let's look at it. So ubiquitination is a three-step process involving three enzymes. So the very first enzyme is called as ubiquitin activating enzyme. So in the first in the first step, the enzyme picks up ubiquitin on itself by using ATP. Okay, with the use of ATP, ubiquitin goes and gets attached to this enzyme. What is the name of the enzyme? Ubiquitin activating enzyme. You should know the name. Now once ubiquitin is attached to the first enzyme, second enzyme becomes active, which is called as ubiquitin conjugating enzyme. Meaning E2, it takes ubiquitin from E1. So as you see, earlier it was on E1, now ubiquitin got transferred to E2 enzyme. And what is the E2 enzyme? Ubiquitin conjugating enzyme. Okay. And then the third enzyme comes, which is called as ubiquitin ligase enzyme, E3. This E3 enzyme, it is already attached to the protein that needs to be degraded. Now I said that the protein that needs to be degraded, it can be called as a substrate and it can be called as a condemned protein. Okay, both means the same thing. So the condemned protein or substrate is attached to E3 already, and then E3 brings about binding between ubiquitin and the condemned protein. And how that binding happens? The glycine on ubiquitin makes a bonding with lysine of the condemned protein. So the protein that needs to be degraded, it has a lysine, it binds with a glycine on the ubiquitin, and ubiquitin gets attached. The bond formed between ubiquitin and between the lysine of the protein is isopeptide bond. Okay, the bond is called as isopeptide bond. Let me go through it once more on the next slide. So as I mentioned, ubiquitination is a three-step process. So what happens in the first process, the very first enzyme is E1, ubiquitin activating enzyme. This enzyme uses ATP and gets attached to ubiquitin. As you can see here, it got attached to ubiquitin. Now, the second step happens in which ubiquitin from E1 gets transferred to E2. The enzyme involved here is ubiquitin conjugating enzyme. And then the third step comes in which E3 enzyme, ubiquitin ligase enzyme works, where it is already attached to the protein that needs to be degraded. It takes the ubiquitin from the E2 and a bond is formed between the protein and ubiquitin. Glycine of ubiquitin makes a bond with lysine of the protein, and that is how the protein gets a ubiquitin molecule. And this keeps on happening, you know, again for the second molecule of ubiquitin, again for the third molecule of ubiquitin, for the fourth, and so on. So the same process, three-step process will keep on happening. Okay? Okay. Now, the protein is tagged with ubiquitin now, but it is not degraded, it has just been tagged. So for the degradation, you need proteasome. So what is a proteasome? What does it do? It degrades most of the cytoplasmic, nuclear and membrane proteins, more than 90% of them, and they are short-lived. Those who do their function and leave. Okay? Also, during degradation, ubiquitin does not break down. It just gets removed and recycled. Okay? And proteasome are large protein complexes. That means they are made from more than one protein. And if I look at their structure, the structure is divided into a core, central core, and a cap region. Looking more into the structure of the proteasome. So if you see here, this one. So proteasome, as you can see, there are so many proteins involved here. It has a central core region. This is the core. And it has cap region, which is the regulatory particle. Regulatory particle. So this that you see here is the regulatory particle, and this that you see here on the other side is the regulatory particle, or you call it as the cap region. Okay? If we look at the core, structure of the core, it is a large cylinder, it is a large cylinder, and if it is made up of 4 rings, 1, 2, 3, 4, okay. The outer 2 rings are made up of alpha subunits, 7 alpha subunits, and the inner 2 rings, the one that you see in blue color, they are formed by beta subunits, 7 each. So alpha 7, beta 7, another beta 7, and then alpha 7. Okay, that's the central core. And this is the main, the core is the main which will do the breakdown. So the core structure does the breakdown. Then why do we need this cap? What is the function of the regulatory particle? If it is not causing breakdown, then why it is even present? The reason that regulatory part or the cap region is present, because it is important for recognition and binding of polyubiquitinated proteins. So actually the central core that you see it does the breakdown, but it does not recognize the proteins that are ubiquitinated. So it's the regulatory part which recognizes those proteins. Also, it is the regulatory part that removes the ubiquitin, because ubiquitin does not get broken down. It is the regulatory part which does the unfolding of the protein. So the protein is degraded not in a folded structure but rather in a linear structure. So the unfolding has to happen, and it is the regulatory part that will move the protein into the central core. Okay? And once the protein has reached the central core, the proteins are hydrolyzed by the central core into small peptides. So, once the protein reaches into the core, it has enzymes, you call them peptidases, you call them proteases, it's the same thing. This enzyme breaks the protein into small pieces, small peptides. And those peptides, they come out into the cytosol as very small peptides. But they are not free amino acids. However, the cell needs to maintain an amino acid pool. Peptides are not needed, amino acids are needed. So what happens? Once those peptides, they come out in the cytosol, in the cytosol also there are enzymes, you call them peptidases or proteases. They further break down these small peptides and release those amino acids. Okay? I repeat the whole thing once again here. So once the protein, as you can see here, this protein gets ubiquitinated, so it is destined to undergo degradation. It needs to go to the proteasome, correct? If we look at the structure of proteasome, it has a core region and it has a regulatory particle on both the side, which we call as cap, correct? The central core, if you see, it has 4 rings, 1, 2, 3, 4. The outer green rings that you are seeing are made up of alpha. How many in number? 7. The inner 2 red rings that you see are made by beta. How many? 7 each. And it is this central core which does the degradation, correct? However, what is the function of the cap region? The cap region is the one which recognizes the protein with ubiquitin present on it. It removes the ubiquitin. It unfolds the protein to become linear. As you see, as it is moving, it is become linear. And it pushes the protein into the central core. Central core has enzymes which break down protein into smaller peptides, as you see here. They are not amino acids, they are still peptides. So in order to convert them to amino acid, they need further breaking down by proteases that are present in the cytosol. Okay? Those proteases will break those peptides and release amino acids. Those amino acids are available now to make a new protein. Clear? Now, regulation of ubiquitination. So, if we look at the whole bigger picture, you must be realizing that the whole process of protein degradation by proteasome is actually controlled by the ubiquitin that is attached to the protein. Because if ubiquitin is not attached, protein will not get degraded. Or if ubiquitin gets attached to the wrong protein, I mean the protein that doesn't need degradation, so a wrong protein will get degraded, isn't it? So the main regulation point is addition of ubiquitins. So ubiquitination is a highly regulated process because it is important for protein degradation. Now for any protein to be broken down by proteasome, several ubiquitin molecules need to be added to the protein. And if you see the last point, and this is important, a minimum of 4 ubiquitin molecules need to be there on the protein for the proteasome to break that protein. Anything less than 4 will not be seen by the proteasome. At least 4 or more than 4 should be there. Okay? However, there is something else which is very interesting about ubiquitination. There are some proteins in which only one ubiquitin gets attached, so that monoubiquitination happens for them. Now with just one ubiquitin they do not get degraded. However, due to attachment of that one ubiquitin, the function of that protein becomes more advanced. So it is like accessorizing that protein to have additional functions. For examples, some ubiquitin, one ubiquitin, if I may be specific, one ubiquitin attached to some proteins can serve other functions, like they might be able to regulate DNA repair by just adding one ubiquitin to such proteins. They are able to regulate transcription, and they might be able to regulate endocytosis, anything that is coming inside the cell. So what I am trying to convey here is that adding one ubiquitin to a certain protein does not take it for degradation, but it can advance its function to do something else as well. And for any protein to undergo degradation, a minimum of 4 or more ubiquitins are needed. Got it, everybody? And finally, the last thing. Now, error can happen. You have seen during DNA replication, translation, transcription, errors happen all the time. So that error needs to be corrected, isn't it? Similarly, for proteins that get ubiquitinated by mistake, that mistake needs to be corrected. And how does that mistake gets corrected? In our cells, we have enzymes which are called as deubiquitination enzymes, DUBs. These DUBs, they are coated by, again this is nice to know information, you don't have to memorize what are the gene families, but just to enhance the process of gaining information. They are encoded by two classes of gene families, ubiquitin carboxyl terminal hydrolases and ubiquitin specific processing proteases. These are two gene families that give you enzymes which are called as deubiquitining enzymes or DUBs. And the name suggests the function. D means lack. So deubiquitining means removing ubiquitin. So what happens, again this point is not as important, we can skip this point. What DUBs do, they reverse the degradation effect by cleaving the peptide or isopeptide bond between ubiquitin and the protein that is going for degradation. So what you see here in blue or grey, whatever you want to call it, is the proteasome. And you see a protein is going inside with, to undergo degradation. So what happens, actually it has two functions, I'll tell you. The very first function is, for example, as I mentioned, a protein got ubiquitinated by mistake. It was not destined for degradation. So these enzymes, they go and they quickly remove those the ubiquitin molecules like this pair of scissors that you see here. They break that bond and ubiquitin gets removed from the protein, and that protein goes back to do its function. Also, these are the enzymes that are removing the ubiquitin from the protein that is destined for degradation also. You remember I told you that the regulatory particle of the proteasome, it also removes the ubiquitin. So it calls these enzymes that please remove the ubiquitin from the protein. Okay? So DUB, it has the main primary function is that it reverses the degradation effect by removing ubiquitin from protein that is not destined for degradation. And also DUBs are also responsible for removing ubiquitin from the protein that is entering the proteasome. Okay? Is it clear to everybody? Yes. Yes, because proteasome will only break down the protein. What this enzyme is doing, it is removing the ubiquitin, because the ultimate tag for any protein to be degraded by proteasome is having a ubiquitin. These enzymes are just removing ubiquitin. They are not breaking down the protein. Okay? The proteins are broken down by the peptidases or proteases that are present inside the central core. Okay? Any more questions? That is all for today. I meet you tomorrow again.